All right, are you ready for more gene expression? I know you are. Let's talk some more. So we already talked about chromosome packing. Now we're going to talk about some of the things that go into deciding which genes get transcribed. And one of those is the presence of these things called transcription factor proteins. I could talk. Okay, so transcription factors are proteins. And they are special proteins that bind together and bind to areas on the chromosome called enhancers. These are what we call upstream from the gene, so some ways along the chromosome. And a lot of enhancers also bind to a region called a promoter. And the RNA polymerase can't bind to the promoter region unless the transcription factors are there. That's why they're called transcription factors, because if they're not there, transcription doesn't happen. So where do these transcription factors come from? Well, it turns out that they are produced or released by the cell in the chromosome, in the cytoplasm, as a result of a signal from somewhere else. So here's a diagram where we have a signaling cell that released some sort of molecule, and that might have been released by, for example, exocytosis. And that binds to a receptor on what we call our target cell, or the cell that we're going to do cell regulation in, or excuse me, gene regulation in. Now, this is going to start what we call a signal cascade, which is one protein binding to another protein, binding to another protein, and finally causing something to happen. This sounds really complicated. It is. It's a bunch of steps. If this sounds interesting to you, it's fascinating. If you can remember it all, go study cell and molecular biology. It's amazing. Please go do that. My brain can't. So yeah, not something I do. Anyway, so we have our signal molecule binds to a cell surface receptor on the plasma membrane. That one then binds to another protein, which binds to another protein, which binds to a transcription factor or phosphorylates a transcription factor or something like that, and activates that transcription factor. The transcription factor, remember, is a protein. It then enters the nucleus and it binds to the enhancer and allows the RNA polymerase to bind to the promoter, and then the RNA polymerase starts transcribing the gene. So the presence or absence of these transcription factors controls whether the gene gets expressed. So in cells that don't ever want to express this gene, well, they can prevent it by just not having the transcription factors, not having the relay proteins, or not having the surface receptors. Remember, things like hormones travel through your bloodstream and bind to receptors on certain cells. Other cells don't have those receptors. They ignore those hormones. So that's one way that transcription can be controlled. Another way of controlling gene expression is in the RNA splicing, which we talked about when we were talking about transcription and translation. So you remember that the exons are coding sections of a gene that will be part of the mRNA that exits the nucleus, and introns get edited or spliced out. Even with the presence of transcription factors, the cell can control which protein gets made by controlling which introns get removed and which get spliced together. So in this case, we had 4 or 5 possible exons, and this mRNA got numbers 1, 2, 3, and 5. This one got 1, 2, 4, and 5. So these are going to make 2 different proteins. So the same transcription factors could result in the production of 2 different proteins. How would the cell decide? Trust me, other proteins are handling all of this. Now, the other thing is, remember that the mRNA has to have a cap and tail. If the cell doesn't actually need the protein to be made yet, can just wait to put the cap and tail on. In the cytoplasm, the mRNA can either stay in the cytoplasm and be translated by ribosomes many, many times, or it can be broken down. So there are other proteins that are going to control whether the mRNA stays in the cytoplasm or gets translated and gets broken down very quickly. One other step is the RNA processing the protein processing that happens in the Golgi apparatus. So this, for example, is insulin, and it's actually made in long strands that contain multiple insulin proteins. So the actual insulin gene has many copies of insulin in it. It gets translated into the protein, the polypeptide. It gets sent to the Golgi, and the Golgi cuts out a section, leaving 2 insulin strands. Those are now the active insulin that can be expelled from the cell, secreted from the cell by exocytosis. So the cell can control whether the Golgi goes ahead and cuts this part out or not, depending on, again, another signal cascade showing whether insulin is needed. And then, does the protein get made? Does the protein itself get broken down by the cell? All of these things can be controlled. And so in eukaryotes, there's many, many layers where we can do this gene regulation, and it does get very, very complicated. So we're going to stop there. That's all the gene regulation we're going to talk about. But in the next lecture, we're going to start talking about how scientists have used these cellular mechanisms to do biotechnology, including some crazy things like cloning animals and creating genetically modified foods.